So I'm going to talk today about, sort of tie in what Scott talked about and what Dr. Valley talked about with regard to molecular analysis in newborn screening. As Sarah said, this is something sort of near and dear to my heart because it's sort of how I was raised. So just to set the stage, as Scott said, newborn screening is basically a population-based risk assessment. It's a screening tool. And the tests that we do in the States have to be universally available, and we have to send out results in a timely fashion. And so the goal of it is to look amongst all of the babies that are born in New York and appear healthy and across the country, and find the one baby who has an abnormality that's not phenotypically present. And so that is the goal of what we do, and trying to incorporate new tests or molecular tests have to fit into this scale. So this slide is from Suzanne Cordovato. When we think about molecular analysis and newborn screening, I'm going to go through sort of what's done today and what the potential is for the future. However, as Dr. Valley also talked about, the cost of genome sequencing, the bang for your buck, so to speak, is pushing a lot of people to consider whether or not whole exome or whole genome analysis would be useful in a newborn period. And he also talked about the clinical testing versus sort of the research test. So the state-of-the-art sequencer now available can produce 16 human genomes in 3 days at 30x coverage, which is pretty good. So in newborn screening in the context of New York State, with 250,000 babies born every day, I would have to have 63 sequencing instruments to complete my workload for every baby. At a cost of each instrument of $10 million, that would be $630 million that would be necessary just for me to buy the instrumentation to be able to screen all the babies in New York universally. People throw around the cost of a genome, and so when they say it costs $1,000 to do a genome test, what they're really talking about is the instrument, the reagents that are used. The interpretation is actually quite costly. So when you hear $1,000 genome, if you multiply that times 250,000 babies, even the reagent costs are way above my budget and many, many other budgets combined. So if we added up a lot of the screening budgets across the country, you would see that most of them would be covered by just that cost, would fall under that cost. So the question really at the end of the day, and Scott just gave a really good example of the fact that in his state, galactosemia mutation analysis really does sort of clear the waters up. But we have the potential to really make the waters muddy as well. One of the things that people say about incorporating molecular testing into newborn screening programs is that we get an increase in sensitivity of our testing. And we also decrease those false positives, those families that we impact by telling them they potentially have something's wrong with their baby and that they need to get further testing. We can reduce that by incorporating molecular testing in a thoughtful way. At the end of the day, the thing you also end up is identifying carriers. Now, people talk about whether that's good or bad to do in a newborn screening context, and you can use them as teaching moments to inform families that there are other people at risk, and potentially that family has a risk to produce a child with that disease. So you get this counseling built in. And people have been identifying sickle cell carriers for many, many years, but with the advent of this additional testing, the volume of carrier detection increases a lot, especially when you go to a sequence-based test. One thing the holy grail of doing this is to be able to predict phenotype. Remember, when you're in a newborn screening context, you're actually looking at a baby who's asymptomatic. It's not like a family who's bringing a sick child and is willing to sort of take what they get, the information that they get from a sequence test. But over time, we might be able to say, your baby has this genetic constellation, and this is what you can expect. Again, that might not happen in my lifetime. The clinician's perception of molecular data is also one thing. We've heard some stories where when we have a baby who has a delta F508 homozygous condition for cystic fibrosis, they say the baby has CF. They don't bother doing the sweat test, which is the real diagnostic. So clinicians' perception of molecular results needs some retraining. And the other thing I'll talk about is timeliness. It's something that we are concerned about in newborn screening. So molecular testing has been around in newborn screening for quite a while. It actually began in 1994 in Washington State, where Mike Glass implemented hemoglobin screening for some of the common hemoglobinopathies. And then and Wisconsin also began just delta F508 testing for cystic fibrosis. And over time, states, sort of on a one-off basis, implemented different tests to help out. So Wisconsin does an NSUD test for a common mutation found in Mennonites. They have a large population of Mennonites, and this one mutation detects those babies. In New York State, we were the first state to begin screening for Krabbe disease in 2006, and we provide full gene DNA sequence analysis for that condition, and then ALD in 2013, Pompe in 2014. And now, essentially, most tests that are being added in this lysosomal storage realm will have sequence analysis. And again, this is because we want to eliminate health disparities, insurance, it will help interpret the biochemical data, and we're able to actually provide that information in a just-in-time basis so that when the baby goes to the specialist, that DNA information is available. The first universal DNA test, although it's not a genomic DNA test, is when Wisconsin implemented SCID screening in 2008. This is a DNA-based test, but we're not looking at a gene. We're looking at a TREC, which is a little piece of DNA floating around in the white blood cells. By 2010, all states were screening for cystic fibrosis, and 40 or more of those were using a DNA second-tier test. So we see the use of second-tier molecular tests to increase the sensitivity or specificity of the primary assay, which still is a biochemical-based test. It helps us to clarify ambiguous results, and it's sort of this just-in-time test, as Scott talked about as well. And now we're moving into the world of where we can maybe implement primary molecular tests as a first tier without the biochemical. And that's good for conditions where there may not be a biochemical assay that's available at this time, but there is a molecular assay. And one example of that is spinal muscular atrophy. You've heard about that in some of the sessions. The potential to pick up those infants. The frequency of that condition is similar to CF in the Caucasian population. And we're able to actually pick up 95+% of infants with that condition at birth by using a first-tier test looking at the SMA gene. A lot of newborn screening programs are also already using molecular testing. This is Centers for Disease Control offers proficiency testing for DNA for several different conditions, and there's 23 countries that were participating last year. So if you take a step back as a program director and say, okay, what do I need to think about in implementing molecular testing? First off is obviously cost. How much is it going to cost? Can I do this assay still within my screening budget? Most states charge for screening, so any new test they add, they have to have either in their budget already, or they have to consider an increase in the fee, and that could be problematic because legislators often don't like to raise any kind of fee universally. What is the value added? Are you actually going to get information out of it that's going to be useful both for the family and the clinicians? Also, the impact on turnaround time. As I said, most tests, all tests that we do today have a biochemical first tier, and then we cull that population down and do the molecular test on a smaller group of babies, not the entire population. But by doing that, you are holding back results for a period of time until that testing is complete. And as you may or may not know, in newborn screening, timeliness is something that we're constantly monitoring to make sure that we get those critical results out the door as quick as possible. The other question, as Scott also said, he had to hire people. Do they have staff who have the proper qualifications and experience in newborn screening program, or do you have to go hire and look for staff to be able to do that work? When we get into sequence analysis, the need for people who know how to interpret the data increases dramatically. So what are the bioinformatics needs, both on a staff and a data storage level? What kind of instruments am I going to buy? What kind of assay am I going to set up? Just the practical workflow issues. And this question of are we screening or are we diagnostic laboratories? There are two schools of thought on that. That screening program should remain screening, and that once you start telling people their genotype, you're becoming a diagnostic lab. So in the old days, back when I was in grad school and doing my fellowship, we did Sanger sequencing by radioactivity. And on a really good day, you could load 96 wells on a gel, and you could get 24 fragments of DNA, maybe on a good day, a couple hundred base pairs. It took all day to do this. It took several other days to get the results. As time moved on, we moved on to this fluorescence-based test. We could do now similar work. The original ones were still gel-based, and so you still had to pour a gel, but the output was fluorescent. And now you could get, on a good day, 800 reads per fragment. And so when the Human Genome Project took off, these large sequencing centers were put into place. And this is the Broad Institute at MIT. And they did this large-scale sequencing to elucidate all of the bases in the human genome. And so now we moved from reading that radiograph and sort of writing down each base to having it printed out to a computer. So big change in technology, and I think if we were still doing the old sequencing, we might not still be done with the human genome. But this really moved things along. And that's still used today. But the next level, which Dr. Valley talked about, is this next generation sequencing, where we have these boxes that could either sequence the entire genome or all the exons in the gene. And now the power has increased substantially. The run times on these instruments can be long, and the prep up front takes a while to do as well. So even though you get more data, it takes longer to get the data. And the output now looks different. So when you think about these jazzy machines that can do all this testing, a lot of times newborn screening programs are viewed as sort of the turtles that plod along slowly behind technology. But in reality, we need things in newborn screening that a nice jazzy molecular lab might not necessarily need. We need cars that are reliable, right? So we need instruments that are reliable. We need to buy many instruments. They have to be, we need redundancy. We need them to be easily repaired. We need them to be relatively inexpensive. So we could have a really fast car that'll get us there quickly and look really cool, but at the end of the day, a lot of times in newborn screening, the Toyota does just well. So when we think about how to implement molecular and newborn screening, Suzanne and I came up with this sort of upside-down pyramid model, where the green is sort of what's the state of the laboratory testing done now in newborn screening, where we have the potential to go, and sort of well into the future. And so I'm going to go through some examples of those, these different parts of the pyramid. So as you heard, Scott had talked about doing galactosemia, and most labs do somewhere between 4, 5 to 9 mutations and provide data just in time to the clinician. CF, similarly, we do a panel of mutations to pare down the risk, the number of babies that are at risk. This is done quite widely across newborn screening programs at this point. When we moved on to sort of sequencing genes beyond a panel of mutations, now we get benefit and we create issues, right? We can do this in a quick way. So in New York State, we do Krabbe DNA sequence analysis, and we still get the results out within 24 hours. So we've incorporated this into our workflow and created an assay that can be run basically in a single workday. So we run the biochemistry first, and then we do the molecular test for babies that have low enzyme activity. And in some limited cases, we can make predictions on the phenotype. So we can tell the provider that this baby has certain gene mutations, which we're pretty sure this baby has infantile Krabbe, and you need to act quickly. What DNA sequencing has done for us is increased or decreased the number of referrals. So these were babies that we didn't have to refer for evaluation. With Krabbe disease, the evaluation is invasive. It's an MRI, it's a lumbar puncture for CSF protein, it's nerve conduction studies. We don't want to subject infants to that unnecessarily. So by doing the DNA test, we've reduced the number of families we need to even call and tell them their baby's at risk. So we've really, in that way, reduced anxiety. But we've also increased anxiety in other levels because we still have to call a fair number of families that are still false positive. So we're looking at ways to make this test even better. Mike Watson yesterday talked about this. Dr. Valley talked about this as well. These challenges of doing sequencing. Again, in the context of newborn screening, we don't have an infant who has any symptoms. We're just sequencing a gene that is associated with a biochemical result. And so the ACMG, Mike showed the paper yesterday, has determined these 5 categories that we use to classify variants. And the known pathogenics are easy. The baby's at risk for this condition. We've seen this in many babies who have this condition, et cetera, et cetera. Benign is also easy. If we know this is a polymorphism that's not going to cause disease, we can release that infant from any further medical evaluation. But the 3 in the middle are what cause the problem when you move into molecular testing in the newborn screening world, because we don't know what to tell those families, and we're not sure how to interpret those variants. And how do you feel when you tell somebody, well, we're not really sure, but you need to do this, this, and this. So it creates that kind of uncertainty, which is not palatable for myself and also for families. But the knowledge that we're accumulating is growing daily, and so hopefully in the time frame that's reasonable, we will have better information. And you heard about a lot of these other areas that are being, other projects that are going on to help with these interpretations. So cystic fibrosis is an example of a test where the first tier biochemical test has very low specificity. So there's a lot of reasons why a baby can have the biochemical anomaly, an elevated IRT or immunoreactive trypsin. That's that first tier test that we do in CF. Those infants then go, states use some sort of cutoff and say, any baby that has an IRT above a certain level will now go for DNA testing. The problem with CF is there's many known mutations that cause CF, and I'll use mutations loosely, many documented variants that are thought and known to cause CF. Most panels that are used by screening programs only look for a subset of those. So now you're left with this uncertainty where you have a baby with an elevated IRT, so you have a biochemical abnormality, and the baby has one CF mutation. Right now, we refer all those infants for a sweat chloride test. And of all the tests we do in newborn screening, cystic fibrosis is one of the ones that parents actually have heard of. But not all the variants that are out there actually cause classic CF. So we'll identify some other types of abnormalities as well. So in New York State, we took a look back to see if we can improve our algorithm. So in New York, we do the top 5% of babies are taken off of the IRT test. And if they're in the bottom 95, they're reported out as normal. And then we do a DNA test for the top 5, and you can have several different outcomes. You could have no mutation detected, 1 mutation, or 2. In the no mutation group, we further parse that out so that any baby in the top 0.1% now gets referred regardless of their mutation status, and I'll show you why we do that in a sec. 1 mutations and 2s are also sent out for referral. So if you're in the 99.9% bottom IRT level, then your result is screen negative. If you have 2 mutations that we're pretty sure the baby has CF, they get referred for workup. In New York, between 2010 and 2013, we had depending on the year, 30 to 40 of those referrals resulted in 19 to 37 cases. The 1 mut babies are more common. So in that time frame, we had 650 referrals, and that yielded 9 to 26 actual CF cases. The very high IRT even gets worse. Out of 250 referrals, 1 to 4 cases are picked up. So in that case, the problem there is shown, I think, on the next slide. The overall yield is 900 referrals in that time per year in that time frame, and then yielding that many cases. So what we did was looked at other tests that were on the market to see whether or not we were able to improve the specificity and the sensitivity of our test. So we used this 39 mutation panel, and we had results that are shown here. So this was over that time frame. In this group of referrals here, we had 22 babies who actually had cystic fibrosis. And most of those babies were non-Caucasian babies. As we went through and so what we did was we looked at a larger panel, and we were able to determine this number dropped because now we identified babies who actually had mutations in the gene. So we wanted to see if we can get even better, and we moved on to the sequencing assay. And this assay now eliminated the babies with very high IRT. We were able to find most babies who had 2 mutations or 1 mutation in that gene. So as we increased the number of mutations, essentially, we increased the sensitivity of our test. So we looked to see whether or not we could incorporate this next-gen sequencing assay into our algorithm. So we start off with the same IRT test and the same cutoffs. We did look at whether we could change this IRT value to spawn the molecular test, and the numbers would be too high. So we kept that cutoff for IRT. And now we're in the process of developing a New York State specific panel, but it's a bioinformatics panel. So what we're going to do is sequence the entire gene using that sequencing assay and only look at the New York mutations that we found in babies with CF in New York from 2002 when we began screening through today. Reveal those first and take a look at those. And any baby who had no mutations, looking at that panel, then we would look at the rest of the gene. We would interrogate all the other bases. And out of those, the babies that have 2 mutations here now would be referred. Same with the 1 mutes. Any baby who went from 1 to 2 mutes when we uncovered the entire gene now would be referred, and the 2 mutes would stay the same. If we use that, we would see that we would reduce the number of referrals on an annual basis from 900 babies down to 100 babies. That's a huge impact on downstream costs. And in New York State, the state covers about half of the births through Medicaid and managed care programs. And so the cost, even though it may not be it might be more expensive for newborn screening, the downstream costs for the anxiety of parents, the health care costs, the time off from work, the travel time, all of that gets reduced. And so we're sort of midstream to doing this. So what we want to do right now is actually target November is to not have the TrueSeq panel up, but look at our current Luminex test at this level, and then move on to the sequencing assay, and then in about a year from now, have this assay in place. So this is a nice summary of what I've just said. The birth rate in New York is about 250. We have about 12,500 babies who have IRTs that spawn DNA testing. The next tier, we would look at those babies with 1, 2, or very high IRTs. And then from there, we would uncover only 100 babies that would have to be referred for evaluation and testing. So that's our goal. So maybe in a couple years, I can report on if that worked. This test actually ends up causing a little bit of an increase in the turnaround time. When I talked to you about Krabbe, I told you that there's no impact on our turnaround time. We essentially, when the biochemical testing is complete, we have the DNA done as well. With this, we're going to actually increase the turnaround time, but we think that the trade-off is worth it for the families, and again, these downstream costs of having to call these families in for further testing. So right now, on a really good day, we get the samples, we do our IRT test, we do the we basically take that and retest it if it's abnormal. We begin the DNA, we do our 39 panel, and the results are essentially done by day 5. This is after we receive the specimen. If we add the sequencing test now, our mailers aren't going to be ready. The reports aren't going to be ready until at best 7 days after we receive the sample. But the number of infants that are impacted are going to be low, so we can release all those other infants prior to and just concentrate on that small population. So the number of referrals would decrease by 89%. The last example of using screening in the setting of using sequencing in newborn screening is looking at SCID. So SCID's an immunodeficiency, and there's many genes that are involved. The immunologists have told us that they provide better care if they know what the mutation, what the gene defect is. And now this is done post-analytically. So the baby is referred by us, goes to the immunologist. The immunologist is the one who actually will order the gene testing. We have a lot of disparity in panels because there's different panels available, And if you're doing things in a stepwise fashion, a lot of times insurance might cover that first test, but they're not necessarily going to cover the downstream tests. So we're able to potentially offer this test and have it as a universal test. So healthcare equality is ensured. So in New York State, we do this TREC test. We look for these little T-cell receptor excision circles that are floating around in blood. And anything less than a value of 125, we refer that infant for a workup with the immunologist. The immunologists then order a complete blood count, a differential, flow, and mitogen studies. And then they may or may not order DNA molecular tests. They've told us they run into a lot of insurance issues and the availability of labs that are covered by people's insurance. So the process can become slow, and the turnaround time for clinical labs for these panels is not a day, it's not 2 days, it's sometimes as many as 12 weeks. So we're moving into months before the baby has an outcome or a result that's useful. So we have a grant from the Centers for Disease Control to evaluate a panel of 39 genes using 2 different next-gen sequencing platforms. And the goal is that we will evaluate this technology and provide our protocols and our experience to other laboratories in newborn screening that might be interested. We're also going to look at whether or not we've actually shortened the time to the final diagnosis. Did the family have fewer visits to the specialist? Was the treatment started earlier? Was it more targeted, et cetera? And obviously disseminate our materials to state programs and provide reference materials to the Centers for Disease Control so that they could offer proficiency testing for these panels. So that's our panel of 39 genes. I'm going to have a quiz at lunch and ask you to write your 10 favorite. We actually just found out that there's a commercial lab offering a 69 gene panel. So, this was good a year ago, and here we go. So the goal of this is we're just going into year 2 of this grant. So we're in the process of finishing up the validations. We've Sanger sequenced. We've done all of that to confirm that what we're finding on NextGen, we're finding on Sanger. And we're going to begin offering. We're in the midst of getting IRB approvals at all the institutions or agreements in place so that we can actually offer this on a consented basis to families, at least to begin with. So when we move down along the pathway, we've talked about sequencing one gene. We've talked about sequencing a bunch of genes that are related to the condition. And you've heard a lot of this earlier, so it was a really good segue for me. Now we can think also of offering testing for genes in a pathway to look at modifiers. You heard that there might be 2 phenotypes at work. There may be genes that modify the phenotype, so the baby may have a gene mutation in 2 different genes that might behave differently than if the mutation was only in 1 gene. We can begin to look at whether or not they impact the onset of the disease, which is a big deal in the lysosomal storage disease screening world that we are living in these days. And then you can also put together a big panel that has only newborn screening related genes. So you get an abnormal biochemical defect, you do sequencing for all the genes that are related to conditions on the panel, but I only look at the one that I'm interested in, the one where there's a biochemical defect. That makes all the testing similar, it makes it more actually streamlined in the lab, and we can manage the data in that way. We have some economy of scale, we can easily turn on, turn off analyses. And we're actually thinking about implementing this in New York and applying for funding to be able to do this kind of work. And then you've heard about the whole genome and exome. And again, be reminded that we're in newborn screening and these babies don't have any symptoms. So all of this information that you get about these variants that are unknown or uninterpretable, you're sitting there looking at a healthy infant trying to make sense of what you're going to, what information you're getting. And so it's not really manageable in a newborn screening setting at this point. So getting towards the end here, the points to consider, the number one thing is, are we going to make it easier for families by doing this type of testing? Are we going to alleviate burden and stress, or are we going to create more burden and stress? These are things that we consider when we move on and we try and do things in a thoughtful way. I've told you about the variants of unknown significance, and you've heard about that earlier. There are variants that are misclassified where people think they cause disease, but after study over a long period of time, they find out that really that variant isn't disease-causing. So classifications are changing with the collection of the data that you've heard about in ClinGen and ClinVar and these other databases. This issue of whether we're screening or diagnostic labs is also hotly debated amongst the newborn screening community. And a molecular diagnosis might not ever result in a phenotype, or it might not result in a phenotype or disease for a really long time. So now you've identified this family at risk, and they're just sort of hanging to see whether or not the baby actually does get ill or begins to show symptoms that then become treatable. And the providers need a lot of education. You've heard about we send the emails about to PCPs and different types of providers about information related to newborn screening and genetics, and we don't get a big response rate. How do we reach them, and how do we relay that information in a way that's useful at the time that they receive it? And also, last night we had a lot of discussion about the availability of genetic counseling and how we can have all of those families seen and counseled for that, for those types of results. But I think we can actually do this right. So APHL has a molecular subcommittee that deals with a lot of these issues and has all of the lab experts represented that do this type of testing in their programs currently. And there's new people being trained as time goes on. So we have some expertise in the newborn screening community, and we have a community of collaboration. We share things all the time. We share information. I think the key is we have to be smart about how we implement this. It would be really cool to have a next-gen instrument and be able to do all this sequencing, but at the end of the day, is it really going to be the best for families? And again, this healthcare equality issue and having that information at time of referral helps with healthcare equality. So I hope I gave you sort of the lay of the land of where we are in newborn screening and where we might be able to go at some point. But as I said, we can't really do this work without the input of a lot of people. So Suzanne is at the CDC. Oops. Oh, I need a take-home message here. Suzanne is at the CDC, and she's helped. We share slides and protocols as well. Mike Glass just passed away this past year. He was the Washington State leader and the implementer of hemoglobinopathy DNA testing in the 90s. Aaron, Denise, they both worked on the CF test along with Colleen Stevens, our center directors, our core at the Wadsworth Center, and really the look-ahead director of the Wadsworth Center, Jill Taylor, who's very interested in having us move along these paths. She's a cheerleader for us within the health department. So, and then people who have sponsored the work. So thank you very much.